Hello, Prekshi.
Um,
Okay. I hope my screen is visible.
yes, ma'am.
Okay. 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 So, welcome to session 8. And today we'll be discussing a bit more about protein and proteomics, followed by a slight bioinformatics and also ethics and research and publications. So, be having three sessions, 40 minutes each of 2 hours. So kindly, once you're done with, we are done with one session, the session will end, and please join immediately in a minute. Okay. So let's begin with the questions, and do feel free to interrupt me and discuss. And I would really suggest only one candidate is present, but I'll suggest that make it more interactive and use your chat box to answer, or maybe you can unmute yourself and answer the questions, whatever you feel is the correct answer, and then we can have a discussion. Let's begin. Okay. Okay. So we have a condition here. The condition is that we are given a set of non-volatile biomolecules and we wish to analyze it through mass spec. So while doing MS-based experiments, we have to selectively choose few sets of ionizers and mass analyzers which are compatible for volatile compounds or non-volatile compounds. So whenever we have been assigned with some compound which needs to be analyzed using a mass spec. So that is we need to decide the sample is very important, that what kind of sample it is and which kind of mass analyzers or ionization techniques we have to use for uh, doing the experiment. So the very first thing is, I hope you understand what is a mass spec, like what in general what a mass spec means, like what happens in a mass spec or what it means. So I would briefly just say that it is a way of analysis in which uh, what we do is we impart some charge to the sample so that it gets fragmented in future, and on the basis of these fragments we detect it. So in a complex mixture of all kind of samples, suppose you have a complex mixture which has different kinds of samples, uh, diseased, non suppose from a human sample, it's from, you take the plasma. So plasma is a source of different markers, biomarkers of different diseases, etc. So what happens is you can identify the components of the sample by using a mass spec. It's extremely high throughput and the range of detection is also very high. So that is how we use mass spec. So by definition, it's an analytical technique in which molecules are converted into ions, which then are separated on the basis of masses to charge ratio. So every ion will have its respective mass, and already we are imparting some charge to it through ionization, and that happens. So what is the type of ionization method in this condition that we would use? Pick the correct examples of such an ionization from the given options. So any idea, Prekshi? Okay. You're saying ionization method, soft ionization, example, mildly good. Anything else?
No, ma'am.
Why not? Why not ESI, and why mildly? And why not both? ASI and MALDI. Both are soft ionization techniques. They are extremely good and they don't they don't actually interfere with the sample, they don't degrade it. Both are good, well established techniques. So why not both MALDI and ASI?
Ma'am, because Maldi is based upon like it is dependent upon uh, like the mass of the of the analysis that we are doing.
Hmm?
So
Uh,
that's why Maldi.
why? What is the full form of MALDI? What makes you think that it's just the mass, and why not anything? Like both of them are ionization methods, right? In a mass spec. So why not both? Like, why do you think that it's only mass associated with MALDI and not with ASI?
Yes,
Any
I'm
guess?
still in doubt.
You're still in doubt. So actually, the
Yes, ma'am.
answer is both. Both.
Okay, ma'am.
Yes, yes. It has to be both. Because listen, the thing is, MALDI and ESI, this particular step of the mass spec, if we like, there are three things in a mass spec. Okay, one is the source of ions, how you are getting the ions, how you're analyzing it, and how it is being detected. Are you there? Hello? Hello?
Yes, ma'am.
I had immediately I had a power cut and my Wi-Fi went off. I'm so sorry for that.
It's okay, ma'am.
Yes, yes. Okay. So, yes. So, what were we discussing? We were discussing that why not both, right? So, what happens in case of a mass spectrometer is, let's just see if I have a picture. Just a sec. Yes. So, you see here, it's not, it's a comparative picture, but it's same. You have your samples here, you have your source ions here, the how they are going to get ionized. So, are you aware of ESI and MALDI? Like, what is the process, how it happens, and how the ions are detected? If not, then I will just briefly say what it is. So what happens?
Yes, ma'am.
Hmm?
Please, like, give me a like brief. Actually, I've just started week 8.
Okay, 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 okay. Then it's fine. So let's begin from the very beginning. Okay, what happens? So what happens is in case of mass spec, as I mentioned, that you are going to give your sample some charge and you're going to fragment it. So what happens is your fragments will have charges and they will be detected on the basis of mass by charge ratio. Okay. So for that, you need to have a source of ions. So why ion, source of ions? Because when we say that we are giving some charge to it, it means that it has to be ionized. So your molecules will be neutrally charged. And then if you ionize it, so it will become some or the other charge will, it will be converted into ions and it will have some charge, either positive or negative, and then that will, now your molecules now have a chart and they also had their own masses, different masses. So on the basis of mass by charge, the detector will detect it. So one thing is very common here, that is time of flight. So this, what is time of flight? That once your samples are ionized, they move through a long column like region. So in that, whichever has smaller mass by charge ratio moves faster, and the ones which have higher takes time, and on that basis the detector detects it. So if you see these uh, chromatograms, so or the uh, graphs that come that after the detector, so what happens is the m by z, this is the m by z ratio and this is the abundance. So which molecule is more abundant, and on the basis of the ratio. So that is how your samples are detected. Now, as I mentioned, that what happens is, because this entire process of fragmentation, ions and different thing comes, so it actually creates a signature or a fingerprint of what peptides are there, what proteins are there. And once your mass spec has already analyzed your data, that data is searched against a known database. Like in the database, all kinds of proteins from a particular organism is there. Suppose you are doing it from a human cell line, okay, and you have done your experiments, so you know what proteins are there in human, that is already discovered long back, and that is in the database. So once you compare it with that, you get to know, okay, so suppose I have a disease condition, these are all the kinds of protein, this is the proteins that are present in a human. In a disease condition, this gets more expressed, so that means that is a reason for the disease. So that's the very preliminary idea of mass spec, that what happens inside a mass spec. So the general steps of mass spec are like, first is you have to generate ions, and that ions are mostly generated in the gaseous state, that we'll discuss right now, that how these are generated. um, yes, so how these are generated. And then these ions are accelerated into magnetic fields. There are magnetic fields which will take them towards the analyzer, which is the time of flight, or the, I just said, a column, like a long column like thing. And finally they are detected on the basis of their flight. Like time of flight means how much time they're taking, or any kind of, it could be a quadruple qt of the different kinds of analyzer. We'll not get into so much of details that what happens. So that that's the idea. So you see the basic difference between MALDI and ASI. Now if we, yes, so there are different ionization techniques. So ionization is the first step. What happens is, on the basis of your sample, so there are different mass specs also, there's GC, there's LC, and there's MS. Okay, we'll discuss this one second. So this GC, this LC, and this MSMS. So GC we do is gas chromatography, when we can vaporize the sample. It can be in gaseous form. So LC is when we can dissolve it, or it is in liquid form. And MS we do when the sample is ionizable. So what are the different sources of ionization? That depends on the type of sample we are doing. It could be volatile samples, or it could be non-volatile samples. For today's discussion, let's keep it to non-volatile samples. So in these two, we have MALDI, that is matrix assisted laser desorption ionization, and we have electron spray ionization. So what is done here, that the sample is mixed with something, or the sample is sprayed in a high speed, so that it ionizes and develops into ions. So let me just briefly discuss what happens in MALDI and yes. So you see here, there's a plate, and there are small wells here. You're there, right?
Yes, ma'am.
Okay. So you see, there are small wells here in the right part of the picture. So what happens is, here you place your samples and you mix it with a matrix. So what is a matrix? It is again a compound which is made up of
Ma'am,
different.
actually, I'm not able to like see the picture.
Okay. So I have not been sharing my screen since, oh, I'm so sorry. Is it visible now?
Yes, ma'am.
Okay. So you didn't see this picture also? Okay. I'll just repeat everything from the beginning. That's best. Okay. So I took up the example of MALDI and ASI. Why? Because the type of ionization technique that we opt for depends on the type of the sample. So suppose today we'll be discussing on non-volatile samples. So there are two types mostly that is done. One is MALDI and one is ASI. That is, MALDI is matrix assisted laser desorption ionization, and that one is electron ionization. So what happens here? So is this picture visible now, the MALDI and ASI?
Yes, ma'am.
Okay, very good. So in case of MALDI, what is done is, suppose the wells you see right here, these are small wells, and you have samples here, like your protein mixtures, complex mixture of proteins. Okay, so you have to mix those uh, with some matrix, which is again a small organic molecule. So once you mix it with the matrix, what will happen is uh, that matrix will help it in the next process. So what is the next process? Then you shoot it or bombard those wells with laser. What happens is once you put it into put a laser or subject the matrix to a laser, the analyte molecules gain charge. So and but the samples will not be fragmented. Okay. So first, what are you doing? You have your matrix, which has small particles, which are again organic molecules. You are mixing it with your samples. You're bombarding it with laser, and then you see the sample is moving up, which now has a charge. So that is how you are ionizing it. This is one process. Another process is ESI, electron spray ionization. So you see a nozzle here. Through this, you pass your sample and it generates into ions because this is done in a very high speed. One. Second, what happens is now because the solution has now the solution having charged molecule passes through the now to need to create a aerosol. So this is a process known as nebulization. So what happens in the nebulization? This area has vacuum. So vacuum is present in the chamber. So the solvent molecules which were around the sample. Now keep one thing in mind. We don't just put in the sample. Okay, the sample is actually mixed with a kind of a solvent, so that now the solvent could be these are mostly organic solvents we do, and the examples are ethanol, so methanol, water, and ACN, like this. So what happens is we mix the sample with methanol, acetic acid, ACN, that is acetonitrile ammonium hydroxide, and depending on the pH of the solution, the molecule will be positively charged or negatively charged, and then we pass the solution through this nozzle. So once this is passed through the solution, is through the nozzle, what happens is it's already charged because owing to the pH. Now we need only the charged molecules and not the solvent. So for that, what is done? Now the process comes that because this region has high vacuum, so the solvent molecules will evaporate, and as a result, only the charged molecules remain here. So you see the covering, or these are the solvent molecules, and the plus minus is the charges. The solvent molecule will evaporate because it is in vacuum, and that leaves behind different charged molecules. So that, and this right hand side is the analyzer. It will enter into the analyzer. Now, to increase the rate of evaporation, what is done is an electrode potential is applied. So there's a difference in electric potential that is applied here, and as a result, now this evaporation process is even more. The rate is increased, and now because there will be same charged ions and as well as differently charged ions, the same ones will repel each other, and as a result, the solvent molecules. So this repulsion also will help in increasing the rate of solvent molecule evaporation. So the idea is here that you're mixing your sample with a particular kind of solvent. So on mixing it, it will give some charge to it. But now, in order for the sample molecules to get rid of all the solvent molecules, we have to give them vacuum and an electric potential. So what will happen when both of these conditions are given to it? It will start evaporating the solvents, and once the solvent molecules have evaporated, the charged molecules will be left behind, and because they are same charge, they could be positive, both positively charged, multiple ions are there, the same charged ions will repel each other, and the rate of evaporation will even turn higher, leaving behind only charged molecules. Now you have molecules which have charged by, you now have given a charge to them because you ionized it. Now they have their own mass and charge ratio. The right hand side, now if you see here, the yes, so these are the droplets, these will pass through it, and then from here on the base, you see the smaller ones which are small mass by charge ratio will go to the detector first, and the larger ones will come later. So that is what happens in case of ESI. So is it clear how ESI works?
Yes, ma'am.
Yes. So now what will happen? The charged molecules will pass through the curved magnets. So here you will see different magnets here. Now that will help in accelerating it towards the top. and there the separation by mass by charge ratio happens. So last time I just and in case of Maldi, what happens? This is your well. You're going to crystallize your matrix with different analytes. So now that you have made, you have different analytes here. So they have mixed. And one more thing, if you're not mixing your samples with your analyte, the laser will not affect it because the matrix has some pi conjugated bonds in it. The compounds which are present in the matrix, they have pi bonds in it, and that actually helps in to absorb the laser, and then it create the ions and move away from the surface. And this process is known as the ablation. So this different charged ions will form, that is known as ablation. So this yes, what happens is, let just recapitulate, or just let me summarize it. So the sample is mixed with the solvent is now allowed to evaporate through the sample, so a dried sample. Okay, now one more important, now because it's a volatile, it's again a volatile substance. So what happens is the matrix is mixed with the sample, and that matrix will be kept to dry it first. Why? So that once it is dried, it will only have the ones which is required. So uh, uh, because the solvent has, again here the concept of evaporation is there. Evaporation is very important here. The solvent is allowed to evaporate from the sample. Why? Because we need only the sample which is coated with the pi conjugated those organic molecules, and not the entire solvent. So we're crystallizing it with the analytes, and then we put it inside the mass spec. Now this plate is put inside the mass spec, and when we are putting laser here, or we are bombarding it with laser on this surface, the ions are generated, which again moves towards the detector and the analyzer. So the laser energy is absorbed by the matrix molecules, and that energetically ablates from the surface of the sample. Now here I introduce one more term, that is ablation. So what happens is that it carries the analyte molecule in the gas phase as well. Like I mentioned, this entire thing happens in the gas phase, be it in ESI or in multi. So this portion is in the gas phase. So this ablation occurs where the sample is we should refer here as analyte molecule is carried here. So from the matrix molecules, now one more thing we need to remember here, that the type of charges that are present here. So MALDI is capable of just positive one charges, but ESI multiple charges are possible. So during ablation, what happens? The analyte molecules are ionized by a proton transfer method. So that's how this occurs. So let's see a small video. Last time also I shared this same thing. So you see that we're mixing the analyte here. Yes, the matrix and the matrix and analyte mixture is here. Yes, we are mixing it, and then we'll be keeping it for drying, and then we put it inside the mass spec, and this is the process of ablation, whether you see the different charged ions are in different colors, they are getting away from the surface. So that is what happens in case of MALDI and ESI. So is it clear to you?
Yes, ma'am.
Okay. Yes. So the answer for this question will be both MALDI and ESI. Okay.
Yes, ma'am.
Okay. Okay. One more thing let's discuss. This is some of the uh, volatile samples. What we do is electron ionization, chemical ionization. These are things I just want to briefly discuss. Is what happens is, so these are also widely used techniques in coupled to GC, like it's gas chromatography and not LCMS. So what happens here is an M plus radical ion is produced that gives a particular molecular weight, and it can be detected. So if you see here, it leaves with a couple of unpaired electrons. So what happens? It loses one electron, and it gets positively charged. because it's losing one electron, and one unpaired electron is remaining. And how the fragmentation will occur, you see A and B, or maybe in this process. So here is the source magnets, how it's collected. uh, well, this is why we need to remember this, because we are discussing here about volatile samples. Till now we were discussing about non-volatile samples, which we are volatilizing it with the help of different organic solvents. Okay, but now if we have volatile samples, electron ionization and chemical ionization processes are used. So this much is enough. Why am I discussing this? Because this third question here, you need this. So it's a match the following. So what happens? The first one, I guess you can answer. The last two, you need a bit help of it. So just have a look into this question. And
B option, I guess.
it's C. Why B? A is gas phase ionization. So gas phase ionization is, till now as we discussed, it will be either MALDI or ESI, because the ionization is happening in the gas phase, right? So A will be 2. And then in case of solution base, that is again ESI, because we are using a solvent and then we are allowing it to ionize in the vacuum. So the starting thing is a solvent. And in case of solid phase ionization, it is generally, uh, sorry, I just, yes, yeah. Yes, so in case of solid phase, Y is matrix, is MALDI, because in matrix associated desorption, the first thing I just mentioned something else, but I saw it is chemical ionization. For A is 2, it's chemical ionization. And for B, for solution phase ionization, it is 1, that is ESI. And for C, it is solid phase ionization. Why are these? Because if you look into the type of sample that we are processing and how the first initial step is happening. So in case of ESI, we are mixing it with a solution, and then we are subjecting it to a high speed nozzle, one. And in case of Maldi, what we are doing, we are mixing it with a matrix, and then we are letting it evaporate and dry. So it is getting crystallized. It's becoming a solid. So the samples are dried samples. So it's a solid phase. So that is the reason behind it. Uh, is it clear?
Yes, ma'am.
Hmm. Okay. So now, likely shifting from mass spec to microarrays. So just have a read, and then we'll discuss like what the question says, and then I'll discuss what it means. Okay. So the question here says that a student from IIT Bombay is performing protein microarray experiments to investigate protein interactors. To detect two distinct type of samples against a printed protein, you have tagged two different colored probes with the samples. As a result, you get a third color, that is maybe yellow, which is definitely not a blank. Given only two types of samples to test interaction, explain what is mostly likely the case. So are you aware of arrays, microarrays?
Not yet, ma'am.
Sorry?
Like I've just started, so I'm just knowing the Maldi little bit and not micro array and everything.
Okay. So suppose you have a chip and there are small locations in it, and each location has a protein in it. Now, if you try to, now you have this is known to you. Okay. Now you have to detect those with the help of an antibody. Okay. So if you put an antibody in it with a detecting system that could be a chemiluminescent, you will be able to detect that area, right? That where it is. So this is a microarray technique. So in the beginning, it was DNA microarray. So what was done is small pieces of sequences of DNA from the genome, from suppose from the human genome, was put in small areas. So now it was probed with your sample. If they hybridized, then they will give you a signal. Now, definitely, whatever you're probing, you will have a fluorescent system attached to it. So, you know what is probing and what is a fluorescent system, all these concepts, are these clear to you? Or hybridization, these terminologies? Okay. So, I'll just briefly say. So, suppose you now, if you have a sequence and you have something complementary to it, so these two sequences will hybridize, right? So that is hybridization. And now we know it theoretically, this is hybridization. How will we understand that actually the hybridization step is taking place? So with any one of the sequences, we will attach suppose a tag to it. Now the tag is a fluorescence tag, so at a particular wavelength it will give a color. So now it's a light, sorry, at a particular wavelength will give a light, a specific colored light. So suppose it is hybridizing. So it is hybridized and now you are able to detect it. So this technique can be exploited to detect many things. Okay. That's okay. Absolutely okay, Prakshi. I'll just, you can use your chat box, no problem. So what happens is, suppose like this, you have a chip like this, which is actually a slide. It looks like a slide. And in different locations, you have sequences here. So now you want to detect that, and you have a blood sample, which is a diseased blood sample. Now you want to detect which particular gene is expressed or which particular gene is present more in case of the disease. Now what are you going to do is you're going to take your sample and you tag those DNAs in your sample with some fluorescent tag, and then you put the samples here. Now whichever sequence it is going to hybridize, you will have a color there. So you're able to know, okay, now this location had this gene, and my disease sample has definitely this gene. So the same thing was used in case of protein. So here you have different proteins here, like one or two proteins, and whenever you have to diagnose something, you will give an antibody which is conjugated to it or a sample. So the first thing is your protein is immobilized to it. Now you have to, from outside, you're going to probe it either with an antibody or with an antibody which is conjugated with another protein, and that is how you detect. So exactly that is happening. So now coming to the question, suppose you have to distinguish two different types of samples. Okay. So what are you going to do? And here, let's be very, like, let's think that your samples, you have generated antibodies against your samples, and you're going to just see if those antibodies are reacting with your protein, these proteins or not. So what will happen? You have two samples and you're given two different colors to it. Okay. Now, and one color is green and one color is red. So wherever you find red or green color here, that means your sample has these two proteins. So whatever was in this location, so these proteins are present, but you're also having one more color. Okay. So what could be the reason? Now let me just make it more easy. One color is suppose control, which is non-diseased, and second color is diseased. So wherever is green is like normal. So even if you're diseased or non-diseased, you will have this protein. So that is a normal, is a control. And red is wherever you are only diseased a protein. So these proteins mean that the particular person has the disease. But also in this plate, you're seeing that different, there's one more color, that's yellow. So what could be the reason? So what does this yellow color indicate? Forget microarrays, forget everything. So what could be the reason? that it's you are also having a yellow color. Blending of two samples resulted in creation of a new sample type. Okay. You're slightly, no, that is not. Like, colors will not blend and give you a new sample type. A new sample type cannot be formed. It's the same, it's two different samples, right? So they are not going to blend because there is no mixing happening here. Because one sample has a particular kind of protein, the second sample has another kind of protein. So when they're binding to some, these are the two probes. So then they're binding to the test or whatever protein is there in the list. So they're not mixing. So it depends that what kind of protein was actually present in the microarray that resulted into this color. C. Yes, yes, exactly. It's C. That the protein had elicited responses in both the sample and overall no differential expression. So what happens is it had same kind of response it had in the both the samples. So because it has responses in both the samples, so wherever the same protein is present, it will give it a yellow color. That is the reason. Good, very. So let's go to the next question. Okay. Now let's discuss this. Imagine you are working in a proteomics lab where you use a sophisticated sample preparation strategy to enrich a specific population of proteins from two different types of lab-grown cell lines that correspond to cell signaling pathway. Now, I have to quantify and analyze these protein cells. What method can I use? You just read a question once again, then we'll discuss. Huh. So, what is happening here? That you're working in a lab, basically in a proteomics lab. So we've used a sophisticated sample preparation strategy to enrich a specific population of protein from two different types of. So you have two different lab grown you have cell lines, and from them you have devised a method so that a specific population of proteins are going to be produced, keeping the rest aside, and that will correspond to that corresponds to signaling pathways. And now we want to quantitatively analyze these protein sets. So how, which method can be used here in the four? SDS page, 2D dyes, fish, or silent? So what is quantitative proteomics? Now, quantitative proteomics is, as you know, it's the terminology suggests it's how you quantitate, or you use quantitative approaches to see the different temporal changes in the proteome, and that can be definitely done by mass spec. So that is quantitative proteomics, and there are generally two methods. One is relative and one is absolute. So relative is like you have a reference, like you will have a control and a disease. So on that basis there will be some differential changes between them, and that can be detected. And in case of absolute, what happens is the exact concentration that will depend on the abundance of your proteins. You will have some internal standardizations, internal standards, and on that basis you will get to know, okay, now this is the on the basis of abundance and internet standards of the you get to know. So that's what quantitative proteomics is about. So what do you think? Which out of these four techniques, which one could be the possible method of detection? C. Fluorescence in situ hybridization. Why do you think it's C? And why do we need to see the localization of the, so as it mentions here, it's like it not only allows to identify, but also localization of biomolecules in situ, which is an improved quantitation method. So why do we need to observe the localization? Why do we need that? So I don't know if you're aware, but fish is actually used in case of oligonucleotides and not proteins. So definitely it is helps in quantification, but in case of nucleotides, DNA, RNA, and not exactly in case of proteome. So here the answer is SILAC, because it's firstly it's a you are working in proteomics, so it's a MS based approach to detect differences in protein abundance among samples using isotope labeling, and it's quantitatively better. Now let's discuss how these things happen. So as we mentioned, in case of quantitative proteomics, we are able to quantitate the changes or what kind of different or what is the quantity, right? For that, we need to have some markers or labels. That how are we going to label it? So one of the labeling techniques is SILAC. So SILAC is stable isotope labeling with amino acid in cell culture. So what is the beauty of this technique is that you are able to you can grow cells using heavy isotopes like C13 or like those. Like you can grow them in heavy medium. So what happens is uh, you will have one culture. Suppose your control is like normal light, and your diseased one or your test one will be heavily labeled. So what is heavy labeled means either the normal will be C12 and the heavy one will have C13. And which compound are you going to label? The amino acids, mostly lysine or arginine. Here you see the picture is of lysine. So what you see, first you cells in light, that is C12 lysine media, you first normally grow them, and then you grow them with heavy. So now the all these proteins or the entire proteome is encoded with these heavily labeled lysine, and then the normal process of you just digest it into peptides, and then you analyze, and this is the difference between 6 Dalton mass shift will be there. So wherever you see a lighter one, is it was the control, and wherever you will see a heavy one, it is the labeled one, is the diseased one. So that's how the difference you can understand, that between if you're labeling two different conditions with two different labels, so automatically the labels will result into different masses, and that the mass spec will easily pick up. So is it clear to you that how this is happening? Okay. So I'll just repeat this entire process, what is happening in case of SILAC. So as we know, okay, okay. So as we know that here you'll see something called light and heavy. We are going to label that we're going to incorporate the non-radioactive stable isotope containing amino acid in the newly synthesized proteins. And so the growth media is supplemented with a heavy labeled amino acids like lysine. The C12 of it is replaced with C13, and in the growth media itself. So whatever protein synthesis will take place and whatever will happen which needs lysine will all it'll all take up the heavy label, like the heavy lysine. Okay, so that is one condition. And another condition, what you're going to do, you're going to label it with light, like the normal one, the normal lysine that is available in the medium. So as a result, what will happen is once you're doing this, the whole proteome is either labeled with the C13 or with C12, depending on the medium constituents. And so now what will happen, that when you take the samples, you digest it into peptides and you analyze. So in case of mass spec, what is very important is digestion, like we digest the peptides. So when we're doing that, what happens is now the peptides are also labeled, the proteins are labeled, everything is labeled with the heavy one, right? And there's one sample which is not labeled. So there will be difference in the intensity, or you see in the peaks. So the light one will be 6 daltons less, and heavy one is 6 dalton more. So you can easily detect the difference between that. So one sample was just normal one with no labeling, and the second one was with the heavy label. So there will be a difference in the shift, the mass shift. There is shift in this, you see in the chromatogram, there's a shift in the mass. So this is detected, and that's how you are able to understand, okay, my this sample was labeled, so it is giving like this, and the proteins. So now suppose this is protein A, and again but this is not present in the diseased one, you see a heavy one in place of that. That means that particular protein is unique to the uh, sample. And again, if you see a change in mass, that also depends that, okay, if it's changed, that means it's getting modified. Okay, less than a minute is left. I will not go to the next slide. Here it will be more clear to you. uh, am I audible? Thank you for rejoining. Okay, so we left here. So what exactly happens in case of silent? So we'll go through the process once again. So the first is the adaptation phase and followed by the experimental phase. So what happens in adaptation phases, we have a starting culture. Okay. So what do? We adopt two different processes, or we process it in two different ways. One is we provide the media with light. It is light chain amino acid, which has only 12C6. And similarly, we have a heavy one. So we see that the chromatogram of these, or the peaks of here. Yes, yes. So you see that here it's a light medium, and here is the heavy medium, the one in red. And we are subculturing as needed, like growing it in generations. And we see that if you see the difference between this and this, the normal peak is here, but the one which is heavy has a higher one because obviously the mass is more. And then we culture it to the number of generations we want, and this is how we get it. Now, now what happens is now the experimental phase. So this is the adaptation phase where we grow under two different conditions. Now this is my control, that is state A, light, and this is my state B, which is disturbed or which was disturbed, or the heavy one was you. Now if we mix it and like fractionate it and obviously digest it, we see there's a difference between the intensity of the MS signal between the heavy and the light one, which gives relative protein abundance between the cells A and B. So in B, you see it's more because it's heavy. And that's how you determine the abundance. Now, if we did not label it, definitely you will have a peak here which is more abundant, but that will not be visible to you. That will not be able to discriminate. So, in order to understand the difference in abundance and the differential experiments, we are opting a process like this. Like in we're growing it in the presence of different labels, and the cells will take up the label, will incorporate it within its protein, and that identification becomes easier. So, is this clear to you? Okay. Last part, please explain. Which part? Experimental phase, which is the last part. Hmm. Oh, it's the same thing that we discussed in the previous. It's actually the same thing. It's nothing different. So now when we are actually performing the experiment, what happens is there's an adaptation phase where you adapt your cells towards this different you're first adapting the cells to different conditions of, be it a high highly labeled or a light chain or a heavy chain or whatever the metabolic tag is, and then you're performing the actual experiment, which is the experimental phase. So you have a control. So this one was your control, which you did not label, or you just grew it with light amino acids. And this one was your test, which you grew with heavy amino acids. So what happens is whatever abundance or the difference in the differential expression of proteins, you'll be able to see it because they're differentially labeled. Now, on differentially labeling it, this is how the MS signal will look. The ones which are non-labeled will look like this, and the ones which are labeled will look like this. So the concept is, now you can ask me, what is the necessary? Like why? So uh, only if you know, if you understand this, that in the entire proteome cannot be detected. Only a very small fraction of it is detected. And even in that, if we start losing our data, then that is difficult. So for that, we use this different labeling technique. So what happens is it is highlighted. When in a group of suppose there's a mass of people who are wearing a white colored dress. Now you want to mark or enrich or specifically highlight few people. So if you ask them to wear some other colored clothes, then you can see them easily, right? That is the concept, that in order to take out or enrich a particular fraction or to have more coverage, we do these different labeling techniques, which were in general, if you ask that in normal scenario, it wouldn't be detected. Now, there are two setbacks of these. One is the amino. What could be the setback of this process? Like, what could be the loopholes of this process? Any idea of Scilab? So, the first thing is you have to have a cell culture for it. It cannot be done on cells or tissues or samples like that. You have to have a growing cell culture, like you will be growing the samples. So, that is very mandatory. And second is, uh, the amino acids, the medium has to be defined. You need to know which media you, that is anyway defined in case of cell culture, animal cell culture. So, but the amino acid sources are need to be known, because on that basis you're going to provide it with that modified hydrogen, or the modified carbon. So that is very important. So that with that we come to end of SciLab. Now there are different other labeling techniques also. So this is SciLab is actually a metabolic labeling, because it's a metabolic tag, right? Now there are different chemical labels like this TMT, this iTrack, this these are the different chemical labels where we chemically label them, and that's a different total thing, the total different process, how we are going to discuss it in details. But there are other chemical tags also, this icat, iTrack, this TMT, and in metabolic labeling there's SILAC and pulse SILAC. And again, this processes, the basis of the process is same, but techniques are slightly different, but we're not going to discuss everything today. And again, there's label free. In label free, what happens is we don't label the samples, we have to process it separately, and just on the basis of spectral counting, that what are the spectral lines, on that basis we get it. So that's how it happens. You see, in case of chemical labeling, there are different these are the different chemical labels. These are your samples, we are labeling it, mixing it, and then we are detecting, sorry, when they're detecting it. So that's how it happens. Okay. So the basic idea is to label their samples, digest it, mix it, digest it, and then analyze it in a mass spec. Yes. Okay. Now let's go move on to the next question, question 5. So let's discuss what happens. Just have a read, and then we can discuss the question. Any idea what will be the answer? So here is a picture of a protein microarray type of protein microarray. We've discussed what is a microarray, how it works. And so like we can just what could be the answer? Yes, Archana B. Okay. Prekshi, what about you? What do you feel? Just look at the image and just see the name. Okay. Many a times you don't need to understand the concept, and you can just guess. Like many a times you will have to guess. You cannot read everything. Reading everything is not possible. There are many a times. Okay. Let's see what is it. Yes, you all are correct. Direct, sandwich, and reverse phase. Can any one of you explain why? Why this is what is the difference between reverse and sandwich, and what is actually happening in this picture? Can any of you describe that? Nice explanation. Direct means no intermediate things. So direct means direct capture. The direct capture. So you are immobilizing your antibodies against your known proteins. You have, you know, these proteins are present, so you raise antibodies against it, and in your microarray chip, you're going to immobilize the antibodies, and these antibodies can bind to different proteins. Now, because you have raised them against known proteins, now whatever, if your if that particular protein is present within your sample, it will detect. So that protein is conjugated with a detection system. Once Okay. Okay, Archana. So once that protein binds to your antibody in the chip, it will give a light, or it whatever detection system is used. So the first one is direct. Sandwich, as you see, you have immobilized this antibody. Your this is binding. Your sample is binding, and to it you are putting one more antibody, and that itself again has a secondary antibody to it. So you're sandwiching your sample between the sample protein between two antibodies, and here is a primary and a secondary antibody. So this is a sandwich. And what is a reverse phase? Here you're immobilizing the antibodies, but in the reverse phase, as you know, it will be the reverse of it. So you're going to immobilize the sample proteins, and not the, you take the sample, you immobilize it, and then you subject it to antibody, primary antibodies and secondary antibodies. If it binds, then it's a reverse phase. So that is what happens in case of the different things. So actually we were discussing about microarrays, and this is exactly what happens. So you immobilize the antibodies raised against the known proteins, and to it you're adding your sample, which could be mostly it's from the blood, like human blood or plasma or serum. Okay. 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 Now, slightly tricky question. So just read it and let's discuss what could be the answer for it. Any guesses for this answer? So if I want to detect a disease, say suppose a newly discovered parasite protein antigen in a patient's blood. So what happens is when yes, we have an answer. B. Okay. So there are few points you need to keep in. No, B is not the correct answer. So you need to actually keep some points in mind. Let's discuss the question first. So what the question says. So suppose there's a newly discovered parasite. So when this parasite will infect humans in the blood, we'll have the antigens or the antigen variants of it. Okay. So now we need to diagnose that what kind of antigen it, and once you're able to diagnose it, so the diagnosis and the prognosis of the infection will be known to us. And this cannot, we cannot wait for months. The person will die if we wait for months. So we have to find a way, find a microarray chip which is fast. First is fast and also cost effective, definitely. So what kind of microarray chip would you develop most likely? What kind? So these are different kinds. So, and also it's written that note that you're supposed to apply patient's blood on the previously printed chip, then wash and finally detected by array scanner. To apply patient's blood on the previously printed chip, then wash and finally. Acha, acha. That means that you have to be using the previous chip, means this one only. Okay. And you need to wash the sample, and then we have to detect it by. Now what do we mean by washing and previously? So uh, generally when we do western, after giving the primary antibody and the blocking agent, we wash it so that the unbound or the non-specific ones wash away and only the ones which are actually binding remain. So that is the same thing it said here. So looking in this picture, which seems a process which can be early, like it'll give you early detection and not late. So just remember one thing, once you're using a primary antibody, you have an incubation period. And again, when you're using a secondary antibody, That's a second level of incubation. So that will take time. So looking into the picture, what do you feel can give you the results fast? Which process has less number of steps, one, and which will give you a fast process? Let's see. A and rest. Prekshi, what do you feel? There's Ayushi also. Okay. Because right now few people have joined. I'll give you a couple of minutes to just go through this question and just settle in and see that if you can answer it. So we are discussing protein microarray chips, and we have to develop, or we have to design a microarray chip to detect an infection, uh, assume most probably a parasite protein infection from patient's blood in an early pattern. So it has to be an early diagnosis. So the question is, if you have these three kinds of protocols or three different designs, which type of design would you select? Your friend Archana says A as a direct, I guess. Prekshi is also towards A, and rest. A. So three people are A. And what about we have more people, I guess. Okay, so yes, the answer is A because it's direct. And you exactly who says it's direct? Wait, yes, Prekshi. Prekshi said it's direct. Yeah. So this is hassle free. You just have to raise antibodies against the infection, the antigen. You place it here. If the sample has that particular antigen, then what will happen? And you are labeling you're already probing the sample with fluorescent, the protein. So if that particular sample has that protein and it's labeled, so that will be detected even after washing. So it's answer A. Let's go to the next one. Okay. So I guess this is the last question from proteomics, and then we will shift to bioinformatics. Let's see. Just read the question once and then we can discuss. Okay. So any guesses? What will be? Oh, ma'am. Yes, Archana. Is this the answer? You feel it's A. Hello tag added to determining the third. How can a sandwich protein microarray detect a third protein? Okay, first let me discuss the question. So you have discovered a protein that influences cancer stage progression. Okay. According to your hypothesis, the protein interacts with non-coding regions of DNA. That's fine. Can you explain Scilab, NAP, and chromatin? Can explain Scilab, NAP, NAPA. Acha. Okay. Yes, I will discuss Scilab. Scilab I've already discussed, so I will be discussing chip. Definitely. Definitely, I will be discussing this one. Yes. So, So my hypothesis, according to your hypothesis, the protein interacts with non-coding regions of DNA while participating in transcriptional regulation of oncogenes. Okay. What are the most appropriate studies for determining if your protein of interest physically binds to the DNA? And how can you further determine if a third protein binds to protein DNA complex of your interest, and pick up the correct set of experiments? Okay. Okay. So now just tell me, I will explain. Definitely, I will explain Silac. But somebody said A. So how can sandwich protein microarray, if we look into the picture, this is a sandwich microarray. How can they detect a third protein? Like you have a protein DNA complex, you have your oncogene, and to its protein interacts. Okay. So, and if there's one more third protein that binds to that, how can that be detected by sandwich? Can this picture, some way you can say that if this picture indicates that a third protein can be detected, is it possible? I guess no. Okay. Now coming to Silat and Napa. So Napa is again one more protein microarray based technique only. So it is a variant of it. And Silac I've already discussed. So Silac is a quantitative proteomics method. In that what we do, we generally metabolically label two different conditions. Like suppose you have a control and a cancer cell line, or a cancer, and definitely you need to have a cell line. Okay. So there we use Silac. What we do is stable isotope labeled. So with amino acids, we label the amino acids in the cell culture. Suppose a control is labeled with light chain amino acid, which has C12, and the diseased one or the treated or whatever is the test cell line, that is labeled with heavy chain amino acid C13. And then we mix them to, and we see the abundance, like what is the difference in protein abundance. So that way we get to know what is the difference between these two cell lines, as well as it could be like cell lines or whatever the protein abundance or differential expression. But here they're talking about cancer stage progression that will mostly come from the protein DNA complex. There are two components here, that is protein and DNA. So that is not detectable in case of Silac. That will not be detected because it's only protein. There we cannot detect the protein. So Silac cannot be done. ELISA also cannot be done because ELISA is a form of reverse phase protein microarray to determine the third protein. No, it cannot be done because ELISA again is an immunoassay based technique, which will, for that we need to raise antibodies against it. And so that the concept of DNA is not here. The very important aspect here is the protein DNA complex of interest. So they've asked us to detect that if another protein can bind to it, and in order to detect any protein DNA complex or interaction, we need to perform CHIP. D, yes, exactly. We have to perform CHIP, chromatin immunoprecipitation. So what happens is these transcription factors that bind to the DNA, that if by some reason we are able to precipitate, so the DNA and protein interaction is intact, and then we are going to fix it and then detect. So we know that these are the parts which are binding to the DNA. So this protein is capable of binding to DNA. This transcriptional factor. Okay. So now let's see a picture how it is done. Yes. So this is your nucleus. It has the DNA. This has the protein. So this is the DNA to which the protein is binding. So it is cross-linked. So what we do is we break it. We shear the DNA strands by sonication. So what will happen is entire DNA strand will break, which is again, it will form the cell lysate. But only the portions which are bound to the DNA, they're not going to break because they're protecting the DNA. And then we add a bead attached antibody to immunoprecipitate the target protein. What happens is now the antibody will bind to this protein and we are going to immunoprecipitate it. So chromatin immunoprecipitation. Why? Because chromatin means the part of the DNA is bound to the protein. So we are going to precipitate it. Now we are going to precipitate it. What will happen is the precipitate will have the DNA, the protein, and the bound antibody. Then we have to unlink the protein and get the DNA out of it. And then we are going to sequence it and map it to the genome. So that is how the particular protein, if it is binding to the DNA or not, this can be understood. Clear? Any experiment which means that, which indicates it has a protein and DNA interaction, always go for chromatin immunoprecipitation, where you have both the players, DNA and protein. I will explain. Yes, I will. This, if I explain this one once again, you will be able to understand. I'll explain once. Yes. So what happens is, here the DNA and the protein is bound within the cell. You are going to perform cell lysis. So on performing cell lysis and enzyme digestion, what will happen? The DNA is going to get fragmented, and so is the other components. But the part of the DNA which is bound to the protein of interest will remain as it is, because that particular, we are not unlinking the DNA and the protein. We are just giving, we are doing sonication with sharing it so that it gets fragmented. Now you see the chromatin is associated with the proteins. This is the fragmented chromatin. Now we are going to precipitate it with the specific antibody. Antibody against what? Against this protein, which is already bound to the chromatin. So now, because this antibody is bound to it, now they will precipitate, and that is the immune precipitate, the chip material. So it is going to precipitate, it is going to get down. We generally centrifuge it. So what happens is it goes down. We are going to precipitate it. It's within the sap. That is what we mean by immunoprecipitation. Or here it will just precipitate. So it will separate out from the solution. The antibody, that is the antibody will bind to it, and it is separated from the rest of the solution. Now what we have to do now, but still the protein is linked to the chromatin. We need to know which part of DNA it has bound to. Exact, right? So for that, we are going to remove the DNA from the protein. That we are going to unlink it, unlink the protein, and the DNA is going to be purified. That is, we could extract the DNA, purify it. There are different processes for doing it. And then for detection of the DNA, what are we going to do? We can do a PCR, we can do a qPCR, we can perform a microarray, RNA microarray, or we can also sequence the genes or the fragments of the DNA to know what exact genomic material was there. So is the process of chromatin immunoprecipitation clear? Or do you want me to repeat it once again? Once again, please. Okay. Sure. No issues. Just give me 2 minutes. There's an issue with my setup, I guess. Don't know why, but is my slide visible?
Yes, ma'am.
And is it moving? I don't think so.
No, ma'am.
don't know what happens sometimes. Okay. Okay. So let's go through it once again. So now the transcription factors actually bind to the DNA, right? So, and we need to determine one such factor, maybe, or a protein that is bound to DNA. So what happens is the DNA remains bound in the nucleus. The DNA remain bound to the protein. There are parts of it which will remain bind to it, and we need to determine which sequence of DNA has this protein bound to it, or it could be also a third protein that is binding to it. So it forms a complex: DNA, protein 1, protein 2, this, that, transcription factor, activator, this and that. It's a complex, right? So in order to detect that portion of DNA which is bound, which is part of that complex, what happens is first we have to lyse the cells. So on lysing the cells, what happens is that enzymes come out, cell membrane breaks down, everything, even the DNA is fragmented, all of these happens, but the part of the DNA which is bound to the protein or the part or the complex remain as it is. Maybe the parts which are not present in the complex will get fragmented, but the parts which are in the complex it remains as it is. As you see here, you have three proteins attached to it, but this one has one. Okay, so like this. So now this is fragmented chromatin. So what happens now? In order to separate this from the entire solution or the lysate, we give antibodies directed towards these proteins, to the complex proteins. So what happens that so that now what will happen? The antibodies will bind to the proteins, and now the proteins are bound to the DNA, antibodies is binding to the protein. Together, these three things will precipitate or will separate out from the lysate. So now on the lysate, you have only your protein of interest, which is bound to the DNA. Now you have to remove the proteins from it. Why? Because you need to determine which DNA it was present, which portion of the DNA it was binding to, where was it present exactly. For that, you will remove the protein from the DNA by unlinking it. Okay, there are enzymes which unlinks it, which breaks the bonds between them. Okay, so what will happen? Once you've unlinked it, your protein and DNA is separate. Now you can detect the DNA by sequencing, by microarray, by qPCR, or by PCR. Clear? So here the microarray is DNA microarray, not protein microarray. Okay, I hope this doesn't confuse you. Okay, clear to all of you? Okay, so someone asked me to discuss SILAC once again. I'll just go back to my slide because this session will end in 6 minutes, and then the last session is mostly bioinformatics, so we can discuss this after that in that last segment of it. So I'll just go back to SILAC and rediscuss it once again. Okay, so you see, this is your how. Now just think that what is done first. So you see, this is lysine. Now in lysine, all the carbons are like 13C. It's generally 12C, but it's 13C. So this is a modified lysine, which you provided in the growth media itself. So what will happen is, once the cells start growing and they start incorporating it through the process of translation, all the proteins that are expressed will have lysine, which is heavy tagged. And heavy tagged by I mean heavy carbon tagged. Okay. So now the whole proteome has this lysine. Okay. 13C6 lysine. Now what? Now your proteome has, wherever there is lysine in your proteome, it is heavy. Now that will, now you digest it. The process of how we process our samples, we digest it with trypsin, and then we analyze by mass spec. Now if you see a difference between the one which was not tagged, the initial culture media, if you do a proteomic analysis from there, and then the present one which was tagged, you see there's a 6 Dalton mass shift. So this is what is actually used here to see this difference, that if you're tagging something, there will be a change in the mass, and that is detectable. The MS signals you will be able to detect. Clear? Archana, is it clear to you? The basic technique of Silat? Yes. So what now, if you are performing an experiment, definitely you're not going to tag everything with a heavy chain, like heavy labeled lysine. So what you're going to do here, so this is your starting culture, which is also this phase is known as the adaptation phase. So you're going to, one media is going to have light, that is the normal one, which may be the control, and the other one will have the heavy one, which will have the 13C lysine. You are going to grow the cells, and once you are grown, then you perform the experiment. What you do, you take the control and you take the heavy one and you mix it. So once you mix it, and then then you have the cell lysate, you do the normal tryptic digestion and fractionation and whatever the mass spec processes are, and you see there's a difference. The intensity of the MS signal between light and heavy peptides give a relative protein abundance between the cell states of A and B. If A and B were your states, so the protein abundance, you can see there's a mass difference, there's a mass shift in the difference. So that is what is done here. So SILAC is like a part of metabolic labeling. It's part of quantitative proteomics and metabolic labeling. So in quantitative proteomics, we label the samples and then we try to detect it using the mass specs. Why? So that more proteins are detected and we can quantify the proteins at different conditions, time points. It could be time points, it could be different drug concentration, treating the cell lines with different drugs. That is what we do here. Okay. Let's go back. Okay. So we'll be shifting to bioinformatics now completely. How can we say metabolism? No, we are not saying metabolism. We are saying metabolic tags because they are incorporated in the lysine. So there's no change in metabolism. No, there's no change in metabolism. You got it wrong. By seeing the change, the entire proteome, suppose there's a protein A, which has which this protein has lysine in it. All proteins have lysine in it, right? Not all. I mean, they have lysine in it. So it's normally tagged. It's as the normal 12C. And suppose you have the same protein, which is 13C lysine in it. Now that will give a different mass to it, right? It's a heavy tag. Will give a different mass. That particular protein, it'll that particular protein, because it's a heavy mass, it'll be mapped in a different way in the chromatogram. The MS signal will be different, the intensity will be different. That is how you will determine the differential expression. It has no effect on metabolism. It is not anything related to metabolism. Just the tag is a metabolic tag. Lysine uptake will be same. Like it, and also if you say about lysine uptake, so in that experiment, now when it was discovered that replication occurs in a semi-conservative way, so there also we had tagged them. We had given heavy hydrogen, nitrogen. That concept was there, na? So the bases had taken up. So uptake is same. Cells lysine be heavy or light, the uptake will be same. There's no preference in uptake, that if it's heavy, it will not uptake. Uptake will be same. Yes. Okay. So is it clear? Okay. So it's just less than 1 minute left. So we'll just Thank you for rejoining. No, this time I'll share my screen and then it's better. Okay. Is the PowerPoint visible? Yes. Yes. Okay. I'll start. Yes. So the question is that you are researching bacterial pathogenesis and you would to learn more about your protein of interest identified and characterized in the same bacteria. Before working with it in the lab, you continue to search in PDB, protein database, and discovered that the protein had no tertiary structure. So you want to use bioinformatics method to create a predicted structure. Which tool combinations are you going to employ? What sort of file will you use as an input? Okay. Any guesses for it? We will perform one. We'll be performing one example. So, any idea? So, are you aware of these tools? SPC, PDB, Blast, Cluster W, Galaxy, FASTA files?
Ma'am, I am aware of protein data bank only.
Okay, only PDB, right? So, in bioinformatics, the beauty of it is whatever you can do experimentally, you can replicate it using informatics, right? So, suppose there's a particular protein structure, it has this many alpha chains, alpha helices and beta chains. You know these and you want to see them. So you can do that in PDB, right? And then you can see it in PyMol, I guess, if you know PDB. And so now here they're saying that you have this particular, but they the protein has no tertiary structure, right? You want to use bioinformatics methods to create a predicted structure. So what is a tertiary and a quaternary structure? So it has actually started with a chain of amino acid, right? That has folded into by using different interactions, and it has developed into a tertiary or a quaternary structure. So these bioinformatic tools are capable of predicting on the basis of different compositions of amino acid. So that is the very brief idea how these two do. Now let's go and I'll just randomly select one protein. I don't know it has a tertiary structure or not, but I'll just randomly select one. Not from PDB, but I will select it from NCBI. I have already downloaded the FASTA file, the protein sequence of it, and then we'll go to XPC Swissprot and see that how the structure is predicted once. And the second thing is that the other option, so the answer is SPC Swissprot. Okay, so the second one, it is PDB and FASTA file. So PDB is more like it gives you the visualization of the proteins and the crystallographic structures and all of these. And FASTA file is a format. Blast and cluster W are mostly used in multiple sequence alignments and to go back to homology sequencing and like alignments and more of evolutionary, how they are evolutionary conserved. Galaxy has a different purpose. So let's just go to Swissprot. And before that, I will just so does how how a FASTA file looks? Hmm. So I downloaded this particular bacterial protein, which is a porin protein. Outer membrane, it's a porin protein of E. coli strain K12. So this is how a FASTA file looks, that it has a greater than sign SP, and this is the so we have to download such a FASTA file, which is also available in PDB. I have not downloaded it from PDB, but I've just for showing purpose. And then we are going to Swiss SPC Swissprot. SPC Swiss mod. Hmm. Start modeling. So you click here on start modeling. It'll take time because of internet speed. And then we have to paste your target sequence or Uniprot KBAC here. We can do both of these. Different formats are of sequences are supported here. You will find it here. Okay. Yes, so it's validated. So you see how they have 90 to 95. These are different. So they are going to predict it on the basis of the amino acid composition, and you see how it. And you can just name a structure. And okay, so we can build a model as well as search for template. So what is a template? Template is a mold on the basis of which another structure is predicted. So this was my previous one. I just tried with it. So just let's go for search for templates. Is it visible to all of you?
Yes, ma'am.
Are you able to follow? This takes slight time. So we'll just wait for 2 minutes. Hmm. Yes. Okay. So you see the templates. These are all the other proteins which are actually predicted. You see there's a different proteins, and here is also the structure of it. Okay. Now you select any one. On the basis of that, you build models. You click on build models. Hmm. So this is your predicted structure. And you can download it, the structure in PDB format, or let's go to structure prediction. Let's go to the summary now. Yes. So this is the model result. Okay. So that is how you build a model of it. Hmm. Clear? How you predict a model here? Okay. Yes. So this is the model that I tried to build up right before, like, I was just exploring. So right now they have predicted just one model, like one model predicted structure, but five were possible using this one template. So yes, that is how it happens. It's how your summary page will look. Okay. Okay. Is it clear that how we process it, how we use it? We need to have a faster file, and then we can just directly paste the file into the query panel and we can have. That is how your structure, predicted structure will look if you don't have a structure already predicted of it. Okay. Now, this is the second question. Uh, should I move on to this or you want more discussion on the previous one? Okay, I hope, I guess you don't need. Okay. So, ma'am, please tell about blast and galaxy. Okay, okay, okay. So, blast is, suppose you have a particular sequence, protein, nucleotide, anything. So, when you want to see which other organisms have the same thing, like how are they evolutionarily related to it, then you're going to perform a blast. So, if we perform blasts using the same query, wait. So I already have I'll just try to perform blast. Okay. Go to NCBI blast. Okay. So this is how the interface looks like. We are going to do a protein blast where we are going to put a one protein and then we're going to search it in the database. So what happens? So accession number, FASTA sequence, you just put it. And database standard experimental, it's new, no needed. Non-redundant organism is it's optional, but we can add an organism, but it's optional. Blast peak. Wait, let's just do a blast and see what happens. Yes. See, that particular protein is present in different kinds of E. coli. We were searching with K12 strain that was of case. You see, everywhere, anywhere that particular porine protein is present, it will show. So how now these are max score E value. The E value is more important. It gets significant the lower it is. So the lower the value, it is more. Now I'm not getting into details how these values are detected and all. So blast is actually used to see that if that particular protein, your query, now whatever you're pasting and you're searching, whatever you're searching against it, it is the you're searching it and the that one is the query. The protein that we pasted. So that query, how much uh, aligned or how much similar it is in terms of coverage or to other organisms, how much it is related. So that is using this 100% coverage. So same proteins are present in, these are generally used to find out evolutionary relationship between proteins, like how a particular protein has generated. It is a paralog, it's an ortholog, how that happens. So that is what BLAST is done. And in case of Galaxy, if we go to the website, so Galaxy is mostly used by, it's to modify your own data when we are working. So you see here, if we click on FASTA queue, barcode splitter, clip, adapter sequence, collapse sequences combined. So these are to edit your own data, but obviously without manipulating your data. So Galaxy is such a tool where you can edit your own data, and it's used by bioinformaticians to, there are different tools. You see FASTA queue splitter, trimmer, joiner, interfacer, de-interfacer. So these are different functions that we need. Or if a particular, suppose you have a file in particular format and you need to change it. Now you have a faster one, you wanted to change it and you don't have that available. So you have to convert it or you have to modify it. That is where it comes Galaxy. Okay. So let's go to this one, this question. I already have the answer. So do you know how to use cytoscape and string? Any idea? So if I want to understand protein-protein interaction in bioinformatics level, like bioinformatically, I want to know I have a protein and how many proteins it interacts with. So do you know how to use it or how to, okay, we'll just perform it. So you have a protein APO A1. We'll go to string database and cytoscape both, and we'll check how it is done. Whenever you are asked to perform interactions, it's always string or cytoscape. Okay. String DB. Okay, wait, just let me go through the slides first and then maybe we can do the demo. Okay, so this is how the homepage of string looks like. You have these different options. You can go for a single protein, multiple protein. You can even try for sequences, different values and rank. Like you saw E value, P value, these are families. So these are how the different things come. So first you need to input the name of the protein and the particular organism. So here it was a human cell line, I guess. So I have given Homo sapiens. You can even put mus musculus or any kind of mammalian organism. And then you click on search. Once you click on search, you will be taken to this page where they'll show you some examples. Like, okay, is this protein? So you see APOB1 Homo sapiens. You select it and you press continue. Once you press continue, you see your APOA1. Where is that? This one interacts with so many proteins. These are the different proteins. And here you can actually see that what these interactions mean. These are the nodes and these are the edges. We say these are the nodes and these are the edges. So what these edges means, what are the color of these edges? So let's go to string DB and check out how it works. Okay, so I'll just yes. So I paste my protein and just let's see what's written in the question. Is it mentioned human cell line or not? Okay, let's keep it Homo sapiens only. You can maybe try later on once the session is over with other organisms. So you paste the name of the protein by name. Then you get this Homo sapiens and search. You get this, select, continue. Yes. And that is how your interactions look, that these are the different proteins it interacts with. Now, Let's go to settings. Yes. So what are these colors? And these are the different colored nodes and evidence like uh so these multiple lines you see it's the meaning of the network edges. What it means that line colors indicate the type of interaction evidence, and if a particular line is thick like this one you see, this means that there are experimental data also which supports this interaction, so it's given. Now higher the confidence level, better is your result. Lower the confidence level means there are chances of errors or false positive. So it's better to keep the standard interaction score that we keep is 0.7, but you will see that here these interactions will decrease, or it may not decrease also. So let's see. You see the interactions, the number of interactions have decreased, the how the way they were interacting. So it's not that they're randomly placed here, they are placed exactly where they're supposed to on the basis of statistical parameters. Is it clear how you see the interactions here? This is one, like one software. The same software is also, this is the web-based interface, but there's a software called Cytoscape. There also you can do the same thing, but in string in this online software interface, you cannot put more than 2000 genes. So we generally don't work with one gene. I there was an analysis where I had to perform the analysis with more than 3000, almost 4000 genes. So I couldn't do string. So then I found that, okay, Cytoscape is also one more platform. So let's go to Cytoscape and see how it looks. So in Cytoscape also, this is the Cytoscape. This is the software, how it looks like. So here you paste the protein. I've already pasted it from before. This you need to select string, the different things. So we have to select string. It's already selected and then enter. Then the search, you see. Yes, this is how it will look. Clear? Here you have more options to play with. Just, yes. These are how, yes, which compartment it is and all the details of the table, like how they're attached to. This is how it's seen. So these are more or less same. Like in string also you saw that all of these were, you could see these. And also I will not complicate you, I will not complicate it more, but there are different other downstream functions that we can check here, that and functional enrichments, that which these genes, which pathways they are present, like which biological processes they are present, which compartment, which cellular compartments they are present in, or also which exactly pathways, cholesterol metabolism, vitamin digestion, fat digestion, you see KEC pathways, reactome pathways. So these are different again web-based sites that we can see which diseases are related to this pathway. So using this, these are very comprehensive interfaces where you can just click on some genes and you can have an idea. So that is how these are done. Is it clear?
Yes, ma'am.
Somebody said no. What is not clear?
Yes, ma'am.
Archana? You're Archana, right? I'm not able to see who is speaking. Archana says no. Archana, what did you say no? Understood, understood. Okay, okay. Actually, I'm not able to see who is speaking. Okay. Hmm. Okay. So the very last question is on bioethics and research community. So the question says that which of the following points can be considered as a serious grievance in the scientific community and is liable for prosecution? Choose the correct combination. Do you have idea of bioethics? Like whenever we are writing a paper or we are filing for a patent, so what happens? Any idea? So any scientific work that is published, there are some rules and regulation. We cannot just randomly we found something and we can publish. No. First, okay. So first thing is, whenever we are doing something, we are on the way of discovering a new thing, we have a start point, right? And that start point comes from previous proven experiments. But we can cite them. We can take reference that, okay, this group has worked till this, and we are taking up the work from here. But we cannot directly copy paste it. That is called, if you're copy pasting something without citing it, it's called plagiarism. Okay. One, this is a very important thing. There are different plagiarism checkers present. After Before submitting a manuscript, generally we submit it to the checker and see how much, because many a times some words will be repeated, and that is accepted. Like less than 10% plagiarism is accepted, but more than that is generally not acceptable by many journals. Okay. So that is one thing. Another is, we cannot manipulate any image. Like suppose I have an image of a gel. I've run a gel and I have the image. The bands are not thick. I cannot use Photoshop to make the bands intense. Okay, that is not allowed. Or maybe there's a pathway. I cannot manipulate and write some other component in the pathway. That is not allowed. Alterations in data. We cannot alter data as it. If we have any data, we have to support it with some proper statistical justification. That I say, the e-value, max score, minimum score, we're not discussing it today, but those have proper statistical significance and they have explanation that why that is important. And the fourth one, we have to acknowledge others' work, because we are starting a work using their help, right? Help, I mean that they have discovered something, they have written a paper, and we are just opting a technique on based on that. So we have to cite that paper. We just cannot say this work was ours or without citing. So these three things are very important. We have to. If a person is found to copy or manipulate or even alter the data, that paper is mostly retracted, and many papers like that are retracted nowadays. Many papers, because people do that foul play here. So that is not allowed, and so that's it. So any other questions for today? Exam is on Sunday. I'm really sorry, Ayushi, but I have no clue about the exam. I just am taking the classes. I have no idea about the exam. Just study the content, I would say. Just study the content. Uh, Try to remember the techniques and the processes, like just we were discussing DNA protein interaction, it's a chip. So try to remember. So some questions you will be able to answer it in that way. So all the best to you. And this was my last session. Some of you have joined all my session. I'm really thankful to all of you for joining my session. It was a very good experience for me, and I hope that I've been able to somewhere or the other share my bit of knowledge that I have. And